All right, this is going to be a very quick one, as we're just going to be talking about prokaryotes and how prokaryotes can become diverse. Uh, Hopefully you remember that prokaryotes do not reproduce sexually. So how does change happen in a prokaryote's genome? Because we know that change in the genome is what causes genetic diversity, and we know genetic diversity is always going to be a good thing in biology. So prokaryotes have to have a method, because prokaryotes reproduce through the process of binary fission. And if you remember, binary fission produces clones, and two clones are going to have the exact same genetic information as each other and this parent cell that they came from. So we need to have some, or prokaryotes, I should say, need to have some way to create that diversity. And the way they do that is through a process called horizontal gene transfer, or another term I could call it is lateral gene transfer. And basically what happens with this is prokaryotes just do a really quick exchange of DNA with each other. The exchange happens through something called a sex pilus. It can also be called a conjugation tube, which is basically just the outer cell wall and cell membrane of the prokaryote create kind of like a tunnel between two different bacterium that get close together. This happens when they get into an environment that is good for the prokaryotes to go through this process. And when the conditions are just right, they'll pass this genetic material between them in a process called bacterial conjugation. We'll sort of take advantage of this when we do our transformation lab later on in the school year. Now, if you remember, bacterial chromosomes they have one chromosome, and it is kind of a circular oval shape, and you have the whole bacteria right here, you got whole bacteria right here. Now, if you look at the bottom of this first bacteria on the left, there's another little small circle here, and that small circle is something called a plasmid. A plasmid is another section of DNA that codes for some very specific functions in bacteria. A lot of times it has to do with antibiotic resistance, which is unfortunately for humans, it can become kind of a problem. If bacteria become resistant to our antibiotics, then they don't respond well. And if they don't respond well to the medications we have, we got to try to find something other way to kill it, or else that bacteria can potentially kill us. So this could become kind of an issue. But basically, the two bacteria will get close to each other, and when the conditions are right, the conjugation tube or sex pilus creates a tunnel between the two bacterium, and a copy of that plasmid goes from bacteria A over here to bacteria B. The sex pilus will then go away, and then you have two bacteriums with, well, they have their same DNA they started with, and now they both have a copy of that plasmid. And the donor bacteria takes that DNA from the plasmid and incorporates it into its own genome, and then they can do everything with that plasmid that the original bacterium A, could do with it. And like I said, a couple big things that plasmids are used for are any kind of special metabolic processes. Anything that one does that another one doesn't do in order to survive, it can now pass that information from one bacterium to another one. Again, these things don't sexually reproduce, so there's no meiosis, no crossing over, any of that. So this is one of the big ways that bacterium can create some genetic diversity, other than having some kind of random mutation, which who knows when that's going to happen. Another thing plasmids code for, again, like I said earlier, is antibiotic resistance, which poses a big problem in medicine, because it can cause problems because we don't have medications that can kill some of these things that we call superbugs. MRSA, methicillin-resistant Staph aureus, is a big problem in medicine right now, because it causes infections that can create sort of flesh-eating resistant diseases that can injure us.